I'm very honored to chair this special Nobel lecture to start off the second day and to welcome Professor Frances Arnold, Linus Pauling Professor of Chemical Engineering, Bioengineering, and Biochemistry at Caltech. Professor Arnold, a pioneer of directed evolution of enzymes to address important problems in chemistry, medicine, and environment, especially alternative energy. Her background in engineering positioned her well to set the stage for what we now call synthetic biology. Professor Arnold is the recipient of many prestigious awards, notably, of course, the 2018 Nobel Prize in Chemistry. Dr. Arnold is a prolific inventor and has co-founded and served on the boards of several companies. Her inspiring life story, as described on the Nobel Prize website, is highly recommended reading and exhibits her amazing drive and resilience. Professor Arnold is currently serving as co-chair of President Biden's Council of Advisors on Science and Technology, and she tells us that she's off to Washington on Monday, presumably for that purpose. So on behalf of the ACA, I'd like to thank Professor Arnold for dedicating her time to serving us, the research community, at the highest level. It's extremely important, and we're very appreciative. And in particular, today, we're honored to have you join us and spend the next hour or so with us. And I would turn the floor over to you and invite you to begin your lecture.
Thank you, David, and thank you to everyone who spends your Saturday with proteins. I have to say this is a real treat for me. I don't think I've spoken to this group before, but I want to start by thanking all of you who've contributed to the data bank, and that's why I decided to meet with you today, because it's played a major role in my research and, of course, in the research of many other protein engineers. Now, that's what I am. I always wanted to be an engineer of proteins, figuring out how to design new ones that would solve human purposes. I'm an engineer by training. I have a PhD in chemical engineering and an undergraduate degree in mechanical engineering. But when I met proteins for the first time as a graduate student in a biochemistry class, I said, that's for me. I want to become an engineer of that world to build better proteins for human applications. Now, this was a long time ago, and that was a pretty bold statement, because nature's done a darn good job at engineering proteins. And the thought that you could make something better than what nature had made was, as I say, it was bold. And a lot of people told me that wasn't going to happen. Well, the point is, though, that nature has not necessarily made the proteins that you want for your application. And I decided to work on the really hard problem, which is enzymes, because as we all know, those catalyze all wonderful chemistry of life, or nearly all the wonderful chemistry of life, and are responsible, for example, from taking abundant renewable resources and converting them into fantastic, adaptable, moving, functional, living machines, sometimes even thinking machines. And nature has done that. Why would we not be able to do that? All right, so this is the early 1980s. So I came out of graduate school and said, I'm going to build better proteins. And as you well know, we had no really good tools then. Everything was hard in molecular biology. Making a single mutation to a protein took weeks of work, and it wasn't clear that it was actually going to work that well, and everything was painful. And of course, most of the mutations that we would choose to make a better protein didn't do what we thought they would do at the level of function. Now, I'm a function person. I'm sorry, structure to me is just a stepping stone to function. And so whether you made the right structure or not didn't matter if it didn't have the function. But let's fast forward 35 years now to today. And we have all the tools you could imagine. You couldn't even imagine back then, but we have the ability to read any DNA we want. Sequencing is almost free. It's remarkable that we can sequence an entire human genome for on the order of $1,000 and soon to drop to $100. We can write any DNA we want. This was really a big advance that you can email off any sequence, as you know, from the PDB, for example, or UniProt, and you get back in the mail the physical DNA in a couple of weeks. That's quite remarkable. And of course, last year's Nobel Prize to Jennifer Doudna and Manuel Charpentier for editing any DNA you want. Remarkable technology that lets you go in and make specific changes inside of a living cell. So we have all these tools of reading it, writing it, and editing. But the problem that I had posed was a different one altogether. And that's the problem of actually composing it, right? So you can read, write, edit, but we don't know how to write it, compose new DNA that has a desired function. And to me, I look at the code even of a single enzyme, and to me, it looks like a Beethoven symphony. And I don't know if you heard some of the conversation at the beginning, but I am neither a great pianist nor a composer. And no one knew how to compose the DNA that would encode a new enzymatic function. So that was the problem that I faced. I said I was going to do this, but I didn't know how to do it. Well, what does an engineer do when faced with that problem? You look for people that have done it. And people, in this case, meant the biological world. There's a really powerful process out there that has given rise to all the function of biology and all of the enzymes that we know. And it's a simple algorithm of mutation and artificial selection. And that simple process, just turn the crank of mutation and artificial selection, has created all the beautiful structures and all the beautiful functions that we know about in the biological world. So I said, okay, let's try the process that's been proven to work and implement some sort of evolutionary mechanism. Now, not a new idea, not a, it's a process that's been used by human beings for at least 10,000 years to engineer the biological world, making things that serve our purposes, everything from corn to carrier pigeons, lab rats, racehorses. We have been modifying biology without knowing how DNA sequence maps to function, right? Just by choosing who mates with whom and who goes on to parent the next generation, we have made things that are not remotely natural, right? This corn would not have been found in the natural world had we not been breeding it. And certainly that poodle is not natural. In fact, it's not even fit. In my neighborhood in north of Los Angeles in the foothills, if that poodle got out, it would be immediately eaten by the hordes of coyotes that go around looking for it. But it serves our purposes and gives us food or gives us comfort, you name it. We have been molding the biological world by artificial selection for a long time. Now, of course, in the past, before we had these remarkable tools of molecular biology, the underlying sequence diversity that you use for evolution was very limited. In nature, of course, worms go with worms and monkeys go with monkeys, but you're not going to cross those species and get anything functional, much less beautiful or useful out of it. You can go out and smoke some cigarettes and maybe dial in a few random mutations, but we didn't have any way to control this diversity beyond a few very limited mechanisms. But that has completely changed. I mean, think about it. We can make any DNA we want, which means, at least in the test tube, we could use 3 parents for artificial selection. We could use 33 parents. We can cross DNA from any species you want. And I'll just point out, Wolford is my laboratory mascot. He's a figment of Photoshop. We do not make chimeric organisms. We make chimeric proteins, but not chimeric organisms. But it's there to illustrate this idea that once you can move to the test tube and you have these remarkable tools of molecular biology, we can start breeding in a completely different way. Now, of course, the problem was, how do you do it in a way that will solve an interesting problem? And back then it was on the timescale of a graduate student PhD, or timescale of getting tenure, right? So maybe a year or two or three for getting something interesting out of it. But before I go into how we implemented it, let's just step back and think about, what is it that you might want to do? So what are we doing? We're freeing these proteins from the constraints of supporting life, right? So now we can explore, using evolution, what is physically possible, including things that have absolutely no natural counterparts. And this bothered some biochemists some years ago. They said, well, proteins don't do that, therefore they can't. But the point is that nature has certain desires and needs that have to be fulfilled by nature's proteins. But once you free it from having to support life, we can explore what they could do. Now, biochemists, and I'm sure many of you fall into that category, structural biologists, generally hold themselves to a very small part of this universe of possibilities. It really is universe size, to those proteins that are relevant to biology, right? They are relevant to solving a biological problem. And so all the proteins that go all the way back to the last universal common ancestor, everything that's ever existed on the planet, fits into a tiny, tiny fraction of that universe of possibilities. The rest of the space is my space. As an engineer, I can go out and look for proteins that will solve human problems. And out there, I firmly believe there's the cures to cancer, there's the solution to the energy crisis. There might even be the cure to death and taxes, for all we know. But we've got this universe of possibilities. And the problem is, how do you get there on a time scale that matters? It really is universe size. And I often point people to this wonderful short story, which I'm sure all of you have read, from Jorge Luis Borges, The Library of Babel, that describes an enormous library of all possible books. And these are books that you could make by randomly assorting, let's say, all the letters of the Spanish alphabet. And of course, in this library is the true story of your life and all sorts of false stories and a whole bunch of books that don't have any meaning at all. And the librarians of this library would throw themselves off the balconies in despair because they never could find anything with a meaningful sentence, much less a whole book of truth. And this is very much like protein space. If you think about all the ways, let's say you start with a protein and you make single mutations, there are thousands of ways to do that. Double mutations, of course, this goes up exponentially, so millions of ways to do that, and you go into billions quickly. And of course, the space of all possibilities is many times greater than the number of particles in the universe. And a tiny fraction are meaningful, and an even smaller fraction solve my problem. So how do you find this and not be like the librarians of Babel, looking hopelessly for your protein? So for me, this problem was, I got a great idea from reading another fantastic short story. And this one was published in 1970 by John Maynard Smith. Another great paper. If you haven't read it, especially students, it's 1.5 pages, and it tells the truth of evolution. And John Maynard Smith, he said, well, sequence space is really big, but evolution, I believe evolution works. And so just from a probability point of view, it's not going to work by taking double steps or triple steps, because the probability of finding functional proteins is very small. But if evolution can walk through functional proteins, taking one step at a time, making one mutation at a time, that's sufficient for evolution to occur. So he basically said, I believe evolution occurs, and therefore I know that the density of functional proteins lying one step away from an existing functional protein has to be at least one out of those thousands. And if that's true, you could walk through sequence space one at a time without falling through the cracks. Now, of course, we know that evolution does make double steps and it does fall through the cracks, but that's not the primary driving force that he was looking at. Now, I looked at this paper, I said, if that's the case, I should be able to turn this into an optimization problem on a fitness landscape where I am now the breeder. Since I'm the breeder of molecules, I decide what the fitness is. That's my application. So maybe I want an enzyme that catalyzes a better reaction on a new substrate, or I want something that's more stable. It becomes an optimization problem where fitness is plotted against all the ways you can change the sequence. And if the landscape is really rugged, then a single mutation will throw you into crevasses of non-function and you stop evolving. And that's what John Maynard Smith said. It's not going to be rugged like that. On the other hand, if it's really smooth, then any fool can optimize on that, because you just close your eyes and take the first step uphill and you can reach the global optimum. So John Maynard Smith's paper made me feel that it's got to be closer to smooth than very rugged. And if it's smooth, you can implement, and these are the tools that were available by the late 1980s, you could implement an evolutionary process in vitro, basically, by taking the gene that encodes for your starting enzyme, and you mutate it by copying it under error-prone conditions. So error-prone PCR was just available. You can make lots of mutant DNAs that way. You can also recombine. DNA shuffling and step methods were being developed. You put that DNA back into cells. They do the hard part, making mutant proteins. And then the hard part for the human being, of course, is deciding who has the properties that you're interested in. And that's called good old-fashioned hard work, analytical chemistry, to measure the fitnesses of individual proteins. But as you know, the numbers are not millions and billions. We're only talking about a few thousand possibilities for making a single mutation. So the trick was to limit the random mutagenesis to primarily single or double mutations. Screen on the order of 1,000 of those. Of course, most mutations are deleterious, so they'll go straight into the trash can in the old days. But if you've define the problem so that a single mutation can give a measurable benefit. And this is really thinking quantitatively about it and understanding what could you expect for a measurable benefit, and can you set up a screen that's sufficiently reproducible that you can see those changes. We were able to just iterate on this and create enzymes that were better at non-natural functions. Oops, let's get this. All right, so what does that look like? It means you take something nature made that's really good at what it does, doing its native job. It's probably somewhere near a local optimum usually. And you ask it to do something new. So I ask it to have a work on a new substrate, for example. It's generally less enthusiastic about that. So the point is, you now turn the crank of mutation and artificial selection and push it up a new fitness peak. Pretty smooth. And that works really well. Now, we all faced a really remarkable result over the last months, but especially last week, where artificial intelligence came in and solved at least some parts of the structure prediction problem. I've been working with artificial intelligence, I call it machine learning, for the last 10 years, because what is the power of this method? So let me just spend a couple of minutes about it, given what's happened recently. Thanks to all the data that you have been putting into the protein data bank, AlphaFold was able to use that to understand the subtle patterns between sequence and structure, so that they could then learn from that data and predict structures, let's say, of a new sequence. I similarly have data, right? We generate protein function data in our screens. There's no equivalent database to the PDB for function, but I do generate data. And so in what I just described to you, pushing the protein uphill, we're taking one path that's smooth in the landscape, and maybe that takes us to a local optimum that's far from some optimum that might be somewhere else. And we throw away most of the data. So for the first 20 years of directed evolution, we threw all the data away. But if you now combine the functional data with the sequence data, and we have new technical methods to basically get all the sequences of the random mutants and directed mutants, then we can build models based on these sequence function pairs. So basically you add a sequencing step to the directed evolution protocol, and you use a model building step. And of course, the most trivial model would be a linear model like linear regression. And our work started off doing that in the 2007 or so. But now, of course, we can have much more sophisticated models, neural net models, for example. And what does this allow us to do? It allows us to do directed evolution. So it's still directed evolution, but basically we can search the landscape more broadly and find better local optima and converge to those higher peaks much faster. It essentially lets us go maybe 3 times as fast or go to higher fitnesses using the same basic evolutionary process. So this is really at the forefront of these directed evolution methods. And of course, at the forefront are other experimental techniques that allow you to get even more data. And what's going to happen in the future is that these will converge, these model building techniques and the experimental techniques, so that we'll be able to do directed evolution very, very quickly. Just give you a little teaser on that. But basically, this evolutionary process works beautifully. And it worked beautifully already in the early 1990s, where we were able to make proteins that did all sorts of non-natural things, like work in a laundry detergent. What self-respecting protease wants to take stains off of your clothes in a laundry machine with bleach and surfactants? And those were all optimized by directed evolution. You can synthesize pharmaceutical compounds with enzymes. All the DNA sequencing is done with polymerases that were optimized by directed evolution in a number of other industries. And for that, work done mainly by other people, right? These are hundreds of application labs that turned to these methods because they were so frustrated with any kind of rational design approaches that I got the biggest accolade of my life in 2019 when I was invited to appear on this set of The Big Bang Theory at Warner Brothers Studios. Do any of you watch The Big Bang Theory? Yeah, I hadn't watched it before I was asked to be on it, but it's about endearing graduate students at my institution in physics, right? Real nerds. It's a whole TV show about big nerds. And for 12 seasons, they went from being graduate students to being a really annoying professors who started lobbying for Nobel Prizes. And now the show is over, and that's probably good news because nobody wants to hear any more about those annoying professors. But one of the last episodes, they invited me and Kip Thorne and George Smoot to appear on this to be lobbied for Nobel Prizes. It was really a lot of fun. I'll just point out, though, after 12 seasons, I was the only woman ever invited to play herself. Right? They had lots of men as cameos, but they never had a woman as scientist or otherwise as a cameo. So finally they solved that. Now the show is over but it's still showing all over the world. It's very popular. All right, let me get back to it. I also got invited to another great party. I have to say, there was a wonderful party in Stockholm where they give you chocolate coins. And Donna Strickland, who's the first woman in 55 years to win the Nobel Prize in Physics, and I got to play with our chocolate coins. Why chocolate? Because Nobel laureates are notorious about leaving their medals lying about, and then they get picked up by somebody else. So they don't give you anything but chocolate until you get on the airplane to go home. But we were pretty pleased about that. And I have to say, it was a great party. If you ever get invited, you should go. There's no doubt about it. It's a lot of fun. Okay, let me get back to science for a little bit. what did we learn from this process, other than getting prizes and things like that? We learned a lot about proteins. We learned that proteins can adapt by this simple process of accumulating beneficial mutations in this simple uphill random walk. Who'd have thought that? And we also didn't know that it would, how many mutations it would take to make a useful protein. Would it take 2 mutations? Would it take 22? Would it take 200? Right? Because most of evolution in the biological world is neutral, right? So lots of mutations accumulate in biology, but they are not functional. They're not adaptive, right? So what is the adaptive component of evolution? And we found, to my delight, and this is why we were able to do it on timescales of graduate student theses in the old days, and now weeks, is that you can get interesting new functions by changing a very small fraction of a protein sequence. And perhaps that's not surprising to you now, right? Because, and especially anybody who studies adaptation in nature, mutations can make a virus more infective, for example, with a single mutation or a few mutations. Now, here's the scary fact that we learned early on from directed evolution. And people had a very hard time with this, but most of the beneficial mutations, not just many, most of the beneficial mutations we would discover were far from the active site of the enzyme, right? They were having a measurable effect on catalysis, but they would be 20, 30 angstroms away from the active site. And that was frustrating to people because we couldn't explain how these effects were being transmitted to catalysis. And of course, they couldn't be predicted, right? So this was left people unsatisfied, and it still does today, right? How do you explain what's happening when the effects on structure are within the resolution, let's say, of crystallography? So there's a lot about proteins we don't understand, but we had this great optimization process. So given that, I want to go to the last section of this, and that's the work that we started about 10 years ago. And that is, okay, let's stop the optimization. Let's stop just tweaking what nature has given us and access activities, enzymatic activities, that you cannot go to a database and find a starting sequence that you can optimize. Chemistry that's completely new to the biological world. Why would you want to do that? Well, the chemistry of the human world is human chemistry. That's the dominant chemistry. We use biology to make wine and beer and a few natural products, but it's not the dominant chemical mechanism. I see a day when human chemistry will be encoded in DNA, and bacteria will do it all for us cheaply, efficiently, and from renewable resources. But for that, we would need to do some transformations, for example, that biology really hasn't cared about. So how do you do that? Wow, how do you evolve a whole new enzyme? And I stayed away from this problem for a long time until David Baker and his designs enzymes really, gave me the impetus, because he'd make these enzymes that catalyze reactions not known in nature. Of course, they were terrible enzymes and needed direct evolution to be publishable, but he could design whole new active sites. And I said, oh, that just burns me up. Evolution should be able to do this. And here's the problem, right? How do you have an inherently conservative process of making one mutation at a time and walking up a smooth fitness landscape to create an enzyme activity not known in biology? Because we think about an active site maybe having 5 functional groups beautifully positioned to lower the energy of the transition state. And it really takes a David Baker to design that. Well, wait a minute. I don't think that's needed, because nature does it all the time, right? Nature is making new enzymes all the time, often in response to human pressures, like dumping potent herbicides like atrazine on the planet. And say for 60 years, atrazine was thought to be non-biodegradable and accumulated in the soil. And then boom, all of a sudden, some organism figures out how to dechlorinate it. This is not a natural reaction, thereby giving the organism a nice, rich nitrogen source. It gives it a selective advantage. And boom, now atrazine is biodegradable all over the planet. How did that happen? Well, I don't have all the time in the world to explain that particular story, but basically it comes down to the fact that enzymes are multifunctional, right? They have promiscuous activities that may not be biologically relevant at the time, but just as a result of their active site, can catalyze other reactions, albeit at a low level. And then that becomes the fuel for further evolution. And I want to send a shout out to my dear friend, Danny Tofik, who, as you know, or maybe if you don't know, he passed away a couple of months ago doing what he loved best, which was rock climbing. And he has done, remarkable work in this area of enzyme evolution, demonstrating how promiscuous, and among many other things, he demonstrated how the promiscuous activities of native enzymes could, for example, as remnants of their evolutionary history. So he showed that enzymes that were related to other enzymes and might have had a different activity in the past and had a little bit of that activity left over, you could use directed evolution to re-optimize that enzyme for its ancient activity, and thereby create a whole new enzyme activity from an existing promiscuous activity. So that's a great idea. And we implemented that recently to create activities you can't find in any database. So let's introduce the cytochrome P450 family. And this is a family I'm sure you know about. There's lots and lots of P450s. It forms a hot oxygen that can be inserted into various bonds in the natural world. And nature has evolved it, presumably through the mechanisms I just described, to do a whole slew of chemical reactions. In fact, chemical reactions that even impress the chemists at Caltech. That's hard to do. But a P450 does chemistry that no human chemist knows how to do. And that's, for example, hydroxylate an unactivated CH bond and at a very specific position, but also can handle this and do other chemistry with that one reactive intermediate. So we said, well, who says that the evolution of a P450 is stopped today? Why can't we take this and evolve it to do yet more chemistry that no one ever thought about or no one has yet reported? So we argued that you should be able to, let's see, let's get this. There we go. That it should be able to, by the fact that it makes that reactor iron oxo species, it might be able to form reactive iron carbene species. This is isoelectronic to the oxo and looks a lot from at least a chemistry point of view, what the P450 actually does. And we know that carbenes are suicide inhibitors of many heme proteins. So it wasn't, a huge leap to propose that the enzyme could form a carbenoid. But the real question was, could it transfer that carbene, just like it transfers the oxine, to another bond? For example, to a silane, an SiH bond, or an SiC bond, and create a new carbon-carbon bond or new silicon-carbon bond. And if it does that, just as a promiscuous activity, could we evolve that? So let me give you just a couple of examples. So we've created a whole new family of enzymes over the last 10 years, which we call carbene transferases. And one of our carbene transferases makes highly strained carbocycles. This is a transformation not known in biology. There is no carbene transfer reactivity known in biology. And also even in the chemical world, it's not known to be catalyzed by earth-abundant iron. In fact, there's only one report with osmium for anything even related to this. So this is really whole new chemistry. But Kai Chen in my group in 2017 and 2018 showed that he could make highly reactive He could make the iron carbonate and transfer it to a triple bond, an alkyne substrate, to make highly strained cyclopropene. So this is like a cyclopropane with another double bond there. And we're talking, almost 60 kilocalories per mole of strain energy. So these are not well represented in the biological world, but the protein is very happy to do this, right? He evolved a P450. He went to the refrigerator and he tested a whole lot of our mutant enzymes for this promiscuous activity. He found the promiscuous activity, and then he turned the crank of evolution and created an enzyme that loves to make cyclopropenes. And we're now supplying gram quantities of cyclopropenes to a whole lot of people who are testing their properties for everything from, polymers to, explosive materials. I mean, they're really interesting molecules, and you can do strain release reactions with them. But why stop with a cyclopropene? If you can add a carbene to a triple bond, you can also add it to a double bond. So you could imagine keeping the cyclopropene in this new enzyme's active site and making bicyclobutanes. Now, bicyclobutane, now we're getting up closer to 70 kcals per mole. That's like rocket fuel. And Kai was able to evolve an enzyme to do that as well. Unless you think that's easy, let me just point out there is absolutely no such activity with a cofactor, right? The heme alone does not catalyze this. And there is no transition metal catalyst that is known to do this. Furthermore, you've got two different transition states for these two reactions. They're by no means identical reactions, adding the carbene to alkyne and adding a carbene to the cyclopropene. So you've got to hang on to the cyclopropene and then have the enzyme active site be able to catalyze both reactions. And furthermore, it's got a short lifetime. So you really do have to, this intermediate that we showed in this particular one is not stable if it gets released in solution. So you've got to be able to hang on to it long enough for this reaction to take place. Nobody would know how to design this, right? If you sat down and tried to design an enzyme that does this, you might as well give up. But evolution can handle it as long as you can find that little itty bit of activity. And very often we find these new activities as we step out into sequence space for new reactivities. And so why is this interesting? Well, it means that you can genetically encode this chemistry in bacteria. So you add your precursors. These are not biologically relevant, but the bacteria will take them and make bicyclobutanes at large quantities, and we can get their crystal structures, and we can show that it's making one stereoisomer of a bicyclobutane, which no human chemist can make. Only an enzyme can do this. Let me give you one more story. The chemistry of the biological world is vast. Right? It's full of innovation and it's really interesting, but it's also very limited. So if we look just at, for example, the carbon X bonds known in biology, carbon loves to bond to hydrogen, nitrogen, oxygen, sulfur, a few halogens, sometimes rarely we see them with a few metals, phosphor, arsenic, but there's whole swaths of the periodic chart where biology, as far as we know, has never gone. And one of those important places is silicon. So there is no known enzyme or compound even in the biological world that has carbon silicon bonds. And that's still true today until somebody actually makes an enzyme that does that. So we took on this challenge. Could we make an enzyme that forms carbon silicon bonds? Once again, fully genetically encoded. And so what do? You go to your refrigerator and you test your large collection of enzymes with a hypothesis. So there is some rational design here, and it's called chemistry. We hypothesize that a heme protein might be able to insert the reactive carbene into a silicon hydrogen bond. So the reaction shown at the bottom of the slide with diphenylmethylsilane, we could form the silicon carbon bond. All right, we tested a lot of things and we found this cool little protein, a cytochrome C, that has no known catalytic function, this electron transfer protein, but it's really, really stable, comes from a hot, salty pool in Iceland. And no, I didn't go there. I went to the databases and pulled it out. And its native function is electron transfer, but it loves to make carbon silicon bonds just because it can. And so 40 times the wild type protein will do it with 97% enantiomeric excess. Isn't that amazing? That's better than any human chemist can do this carbene insertion reaction. And here the protein was just waiting for you to discover what it could do. All right, I had to show a structure here. We actually got the structures of the carbene in the active site, which I won't share with you because it's pretty, I've got a limited amount of time, but I will tell you a little structural story. When we talked about this work in a talk before publication, a structural biologist who really knew a lot about these cytochrome Cs jumps up and says, well, wait a minute, Francis, there's no active site in a cytochrome C. So how is this chemistry happening in the protein? And in fact, if you look at your structure, which it had already been deposited to the PDB and really nice resolution, and you roll your little computer ball over the active site, in fact, you come up with a big fat 0. And, you'd say, well, gee, how does that happen? But this is the lesson that you get from evolution. Nature does not care what your calculation says, because you're not looking at the properties of the protein that are relevant to this system. And the properties of the protein that are relevant to this system is that not only does it catalyze the reaction, but it evolves. So it has a little bit of activity, 40 turnovers in the wild type protein. And as you make more mutations in the active site, you can rapidly increase that activity to where we got catalysts that do it thousands of turnovers, which is really very remarkable, much, much better than anything that a human being had reported for this kind of chemistry. And again, it does it inside of living cells. So inside of bacterial cells, you can now add these non-biologically relevant substrates and form new carbon silicon bonds. Now, this is a funny publication story. When we talked about this work, this is at the same conference, there was a reporter from Science sitting in the front row, and he goes, oh, this is so interesting. I want to write a story about it, but you know very well that you shouldn't publish by press conference. So I said, nope, I have to zip my lip. I'm not going to tell you anything more yet until the paper's actually published. So Rob Service wrote this great article in Science that we're taking steps towards silicon-based life. I said nothing about silicon-based life. I said I made bacteria that can do it, but silicon-based life is something that interests a lot of people, apparently. And you might see Captain Kirk and Dr. Spock in the Horta episode of Star Trek. They're trying to commune with the silicon-based life. Well, that really triggered a lot of interest in chemistry for the first time. Of course, no one read the paper when it was finally published because it had 130 pages of SI and 20 new silicon carbon compounds, but it was really boring chemistry paper. So instead of reading the paper, they all read this article. And the news went all over the world when the paper was published, and I got, called out by Popular Science saying, well, silicon-based life, I don't think so. And there's really good reasons to think that silicon-based life would be very difficult on this planet. But I didn't even get into that debate. And then Astrobiology magazine, of course, loves the idea of silicon-based life. And so all 17 readers of Astrobiology magazine clicked on this article, making it the most read article of 2017. And then the Evolution News, the creationist and intelligent design community calls out and says, oh, this has got to be intelligent design. And I said, yes, evolution is definitely the intelligent designer. But for all of you who really love to see the humor in your science, put it out on Twitter. Okay, that's my favorite. You get real reviews of your science on Twitter, including some some that had us rolling on the floor laughing about how people viewed making carbon silicon bonds. I won't read them because they're a little bit off color, but they were really fun. I'll just point out though, we were able to, in just 2 years, add 2 whole new elements, not just silicon, but now boron as well. to the periodic chart of genetically encoded chemistry. And that's huge, because you think about silicon, there's 50 products in the room that you're sitting in right now that are carbon silicon bonds. That's a huge industry. Siloxane industry, you know, in your hair gels and your earbuds and caulks, sealants, paints. It's an entire industry, multi-billion dollars, mines millions of pounds of platinum every year, hundreds of millions of dollars worth of platinum at huge environmental costs. You know, it'd be wonderful if we could implement silicon chemistry in bacteria. I hope to live to see that day. But you know, it's fun to think about where biology could go with just a little bit of assistance. Now, I could give a whole other talk, but I won't, on making. So I've talked about making carbines and transferring carbines. You can do the same trick with nitrines, right? This is not a one-trick pony. The heme proteins can form reactive nitrines, and now you make new bonds to nitrogen. Very, very exciting, and a lot of potential applications, but that's for another talk and on another day. So I want to end with this idea that, you know, we all talk about the Internet of Things, and that's an exciting area to think about how all our little machines can talk to one another. It doesn't hold a candle to the Internet of living things. The biological world is constantly adapting, sharing DNA, recombining ideas, and basically problem solving by crowdsourcing for the last almost 4 billion years. We have a lot to learn from nature on how to build new things and also survive into the future. So the next billion years. So with that, I want to thank the remarkable group of people who've worked with me over the years on evolving new enzymes. This is kind of an old picture from Nobel celebrations, but it's very lively, and too many people to name and remember, but the students do all the work. I just go around getting credit for it. And thank you for attending.
Well, thank you very much. That was incredibly inspiring and thoughtful talk. Appreciate that. We do have some time for discussion, and there's already a couple of questions I see in the chat, but do feel free to unmute or raise your hand if you'd like to interact personally. Would the people who put questions in the chat like to unmute and ask them themselves? Or shall I just go ahead and read them? I guess I can hear a very faint sound. Can you increase your volume at all? Oh, there. Try again.
Okay, I was hearing. Can you hear? Yeah, I had misprinting. So apparently life occurred on Earth within 250 million years, approximately, from nothing to cells. Do you think this time is enough, or we should suspect that prebiotic life occurred on other planet and was delivered to Earth by asteroids? Something?
So, origin of life, I know nothing about, right? I'm talking about evolution of proteins. So after proteins already existed, origin of life, to me, that's almost like religion, because there's so little data to really go on it. So I don't speculate about the origin of life or the evolution of complexity, which is an incredibly interesting field. How do you go from how do you create proteins in the first place? That's origin. And some really smart people, including Danny Tofik, had been thinking about that problem. But I take it, once it's made, how do you go out and radiate into the future? And other people are working about how do you now radiate into complexity, but that's not my field. So I think it's best that I not speculate.
Thank you.
Dr. Oren, is that right?
Hi there. This was a wonderful talk. Thank you very much. I am actually a graduate student of your brother, Eddie. So it's really nice to see you. I was wondering if you think that one could actually solve the global warming problem with directed evolution. Is there a way of fixing the carbon dioxide problem on Earth?
So I don't have any particularly great ideas, but there are a lot of people working on biological systems that can fix carbon in novel ways. So Tobias Erb, for example, at the Max Planck in Germany, has whole new, and he works with Ron Milo from Israel, and they've established whole new metabolic pathways for fixing carbon. Whether those will be competitive, right, with other methods, more physical methods, for example, I really can't say, but we need every bit of creativity there that we can have. And so the nice thing about directed evolution is that as long as you get a little bit of, as long as you make some lousy enzyme, you can make it often, you can make it a lot better. So it pulls us out of the weeds, right? But getting that lousy enzyme sometimes is really hard.
I don't see any other hands. Anyone else? This is a wonderful opportunity to have a great discussion. Well, I guess I'll jump in and then take the prerogative. So just sort of a sociology question, I guess. I mean, we've seen, you've described a lot of new products, if you want to think of them that way, or reactions. And we've seen in the last couple of years the advances that have come out of biomedical research and even structural research into the pandemic issue. And I think one of the things that we've encountered, and we were certainly aware of this before, was it's all very well for us to spend our careers making pharmaceuticals or vaccines or whatever it is, but to actually get uptake, to actually get them into the community or where they need to be, is obviously becoming quite a challenge. How do you see that whole side of things?
Oh, well, that's a lot of what's going on in Washington these days, is asking, how do we support science so that products will come out of it, right? So the answer to that is, first of all, you have to support science and basic science in the first place so that products can come out of it. But then you also have to support the whole chain of innovation. As an engineer, this is something I've done my whole career. I start companies. I talk poor graduate students into doing all the hard work of starting a company. But if I say, look, it's possible you can do this, sometimes they do it. And that's where the transition from your fundamental discoveries into applications will go. And there needs to be some support along that chain. So I think you're going to see, well, the number of bills coming through the Congress now that will increase the budgets of our science agencies in basic science, but also especially in translational science areas.
I guess what I was sort of getting at was, even if you have a successful product, for example, a vaccine is the thing at the top of our minds at the moment. How do you get acceptance? Like there's large communities that will not accept it, even if it's formed on the foundation of good, solid science.
Yeah, so trust is a big thing, right? Scientists have made some big missteps. We have some blame there, and certainly our leadership has made huge missteps. The previous administration just really undercut the messaging that we needed to pull our country together on the vaccinations. But you're never going to get everybody to believe in it. And that is a problem. In my particular area, I do a lot in agriculture, and we've got to feed 8 billion people soon. And that's going to be really hard to do without genetically modified plants. But we have a big communication problem for solving those. So scientists have to get out and demonstrate the benefits to human beings.
All right, I have a question. I put it in the chat, but these aren't coming up. Dr. Arnold, this is not political, it's scientific. You're teaching bacteria to want to make carbon silicon bonds. How do you apply evolutionary pressure to these bacteria to make them evolve to do that better?
So I screen, right? It's just like the way you would breed cats and dogs. You know, I make the mutant enzymes in the bacteria, and then I screen the bacteria one at a time to find out, one mutant at a time to find out who's doing it better than their parent. And then you take the genetic material and you just iterate on that. So it's not a selection. That's really the important thing. It's not a selection, it's a screen. And because it's a non-natural function, you can't do a selection.
Got it. Thank you.
A lot of people think you can do a selection. They spend 4 years trying to figure out how to get bacteria addicted to silicon. And by then I've solved the problem.
Anyone else want to take the opportunity? I see Wayne is video is on. I don't know, Wayne, if you wanted to ask anything or no. You just want us to see your face, I guess.
So David, Stephen Burler here.
Yes, Stephen, please.
Francis, as the current director of the RCSB Protein Data Bank, I want to thank you for sharing your story with us and for making so evident the importance of open access to 3D structured data, starting all the way back in 1971. With the first 7 structures to be placed into the PDB archive. Thank you.
Well, thank you.
So we do have a couple of minutes, and I see Samantha has a hand up. Samantha, would you like to unmute?
Sure. Hi, thank you for a very interesting talk. So I was just curious because I know all of your systems, you're doing the evolution in bacterial systems. So have you looked into any kind of equivalent directed evolution with post-translational modifications, which obviously you would need to shift to a different kind of system, but with post-translational modifications, they also influence so much of the function of things. So have you looked into any of that?
So we have looked into yeast. Yeast will over-glycosylate, but there are a number of enzymes, especially in the energy area, so cellulases, where the glycosylation is at least somewhat important. So we've worked on evolution in yeast. But as you go to other systems, it becomes technically more challenging. And I leave that for many other groups that have even established directed evolution in human cells. So it's getting better, but it's still hard.
Yeah, definitely more complicated. Thank you.
Chris, go ahead.
Yeah, just have a quick one, Francis. I just wonder, in all the things you've done, whether or not in terms of the sequence space of proteins, whether typically you find it sufficient to just stay near something that already exists or already works and then do direct evolution away from that? Or do you ever feel the temptation, or have you done sort of larger jumps in sequence space where you go far out to sequences which are quite different and then perhaps do something that's also useful or more useful? Don't know.
So the answer is yes. So people like David Baker, who make designed enzymes, right, that don't exist in any database and can't be, identified as being related to anything, he uses directed evolution once he's made these, novel enzymes. So that's an example of where that's been done. I do it by recombination. So for example, I can do recombination and get something that's, 20-30% different from anything that's in the database. That's generally semi-interesting. It's really interesting from a patent point of view. If you want to get around somebody's patent based on sequence, it's a great way to do it. The real question is, do you get functional diversity by recombination? The answer is yes, too. You get some, it's like you and your mate get together and you have a bunch of children, they're not identical to you, right? And you see that with proteins as well.
Right. Yeah, we've seen some of that. Okay, thanks.
Terrific. Well, we're just about hitting noon Eastern time here, so I think that we'll call the session, this part of the session over. But Francis, thank you so much for taking the time and absolutely fascinating discussion.
Thanks, everyone. It's a pleasure.